Hi everyone. Today's lecture will be on multi-omics integration. So I'm Eden. I'm a data science analyst in the Mayan lab. And so just to overview what we'll be talking about today, we're first going to just go over the different types of omics. So there's many of them. And so just to make sure we're all familiar with what they're measuring, and then maybe some strengths, limitations, and unique features of each omic. Then we'll talk a bit about the some tools and resources that are out there for multi-omics analysis, and then most of the talk will be around these data integration methods for multi-omics. And then lastly, we'll have these three examples of multi-omics analyses that kind of demonstrate some of the data integration methods we'll discuss. Okay, so first, just what is multi-omics? So multi-omics kind of comes from this idea that we can observe the same biological system at many different levels. So for instance, we start out with DNA, which is transcribed to RNA and then translated into protein. So at each stage of this process, how genetic information or biological information is transferred through the cell and leading to some organism's phenotype, we can observe the molecular products and processes that are happening at those levels. So for instance, we might quantify genetic variants or the presence of genetic variants in the DNA. We can observe what that looks like at the transcript level, so which isoforms the transcripts are expressed, and then how those are then translated to amino acids and then structured into proteins and the function of those proteins. So there's just from bottom up, I guess is how I've described it, but from at each of these levels, there's a lot of information to observe. So traditional single omics analyses, we look at these one by one. And then multi-omics refers to the integrative capture and analysis of these cellular processes at all these different levels. So in the past decade or so, this has become especially relevant because of advances in high throughput technology. So it's now much easier and faster to capture many of these different omics from the same samples. So what's been demonstrated now through some studies that have used multi-omics analyses is that if done properly, integrating information from multiple omics often gives us much richer and more accurate insights into the biology than what we might glean from just looking at one omic. Okay, so we're kind of just going to walk through some of these different omics from the bottom up. So the first one is genomics, which broadly speaking looks just at the complete set of DNA in an organism, including not just its structure and its function, but also maybe how it evolves and relates to other organisms over time. And out of all the omics, genomics definitely been around for the longest. And so as a result, it has some of the most well-developed technology. And in terms of capturing that genetic information, next generation sequencing has been instrumental in getting us, like comprehensive, large-scale genomics capture, because it took us from getting, just a few sequences or mutations at a time to characterizing like the whole genome of an organism in a single experiment. So for genomics, how it differs from many of the other omics is that the vast majority of the genomic information is actually identical when we're studying individuals of the same species. So for humans, it's like more than 99% of genetic information is the same. And then the information that is usually going to be of interest to us when we're trying to understand diseases or biological conditions are those variants, which make up maybe even less than 1% of the genome. So with next generation sequencing, because we've sequenced the entire genome, we essentially don't have to know which variants we're looking for yet before we can just go in and find them. So we can identify those smaller sequence variants where maybe it's just the difference of a few nucleotides, as well as those larger structural variants like copy number mutations or gene fusions. And then in cancer research, this is especially relevant because we know that tumors are driven by these somatic mutations that accumulate and some lineage of cells over time. So I guess that's one important distinction to make when we're discussing genomic data and variants, is that depending on the goal of what you're studying, so if you're trying to study disease from a hereditary perspective, you'll be looking at germline mutations, whereas somatic mutations are more relevant for specifically cancer and then other diseases of kind of more of an acquired origin. Okay, so one layer above genomics is epigenomics. And here we're observing all the modifications that are made to DNA structure and accessibility, not to the sequence itself. And unlike genomics, which, as I mentioned, is mostly the same between any two cells at a given time, epigenetic marks can differ between cells and tissues. And not only that, they change over time, albeit relatively slowly compared to some of the later omics we'll discuss, but they'll still, like methylation, acetylation, these are all processes that are still dynamic. So when we observe things like DNA methylation or histone acetylation in one sample, something just to keep in mind is that those changes are likely not happening uniformly across all the cells in the sample, especially if you have different cell types or different microenvironments that are represented in your sample. So epigenomics, it really has to do with the way that DNA is packaged in the nucleus and then accessed for transcription by transcriptional machinery. So in a transcriptionally silent state, like in this picture on the right here, DNA is coiled up really tightly around these histone proteins. And so they're stabilized by these like strong ionic interactions between the DNA molecule and the amino acid residues. And what that tight packing does is it prevents DNA binding proteins and other transcriptional regulators from getting in there and then transcribing the DNA. So epigenetic modifications control this basically by loosening up the DNA around these histone proteins, or maybe modifying the DNA itself. So methylation is one of those to kind of prevent the transcriptional machinery from getting there. So one of the most common ways of measuring the interactions between those regulatory proteins and the DNA sequences themselves is this chromatin immunoprecipitation sequencing method, ChIP-seq, which basically it couples the protein precipitation of the chromatin, so DNA and protein precipitation, with next generation sequencing. So when we put those together, what we end up with is we know the proteins are present in the sample, and then we can map them to the DNA, the specific like nucleotide to nucleotide DNA sequences that they bind to. So this has been really valuable in building transcription factor and target libraries. So ChIP-seq has been around for a while, so we have relatively comprehensive knowledge of or characterization of how these regulatory proteins are interacting with the genome. And then another type of epigenomic technology that's common is it's a genome-wide chromatin accessibility characterization. So ATAC-seq stands for Assay for Transposable Accessible Chromatin Using Sequencing. So this method differs from ChIP-seq in that it is agnostic to the specific proteins themselves. So we won't know which transcription factors or proteins are binding to the DNA, but we can get a general kind of comprehensive picture of how accessible the chromatin is to those regulatory proteins. And generally, the ATAC-seq is like faster and simpler to do than ChIP-seq. And then, so the last modification or sequencing method is bisulfate sequencing. This is specific to DNA methylation, and methylation is very important in a lot of diseases. So it gets sort of its own ohm called the methylome. And yeah, so what methylation does is attaching that methyl group to the cytosine of the DNA molecule. It kind of just acts as a blocker for the binding of any regulatory protein to that promoter sequence. So highly methylated regions are transcriptionally repressed. And methylation is one of those reversible modifications that changes relatively quickly. So there's a lot of interest in looking at how these methylation changes contribute to disease. I think, especially in cancer, we know methylation of certain sequences is known to be like a driver for certain cancer-related processes. And then, so yeah, with bisulfate sequencing, essentially it's a technology that turns all of those methylated cytosines into a nucleus. So it turns all the methylation differences into nucleotide differences, and then we can use our next generation sequencing methods to get that full genome-wide sequencing of or characterization of the methylome. Okay, so above epigenomics is transcriptomics, which deals with the complete set of RNA products that are transcribed from the genome under specific conditions or in a specific cell. So like with epigenomics, transcriptomics are going to differ between different cells and different tissues. So the most widely used transcriptomic is RNA-seq, which is this, so we amplify all of the messenger RNAs and then count them. So the measure we get from RNA-seq are read counts. And I think in one of the lectures we've talked about how we process and normalize and then analyze that data. But with RNA-seq technology, this can also be applied not just to messenger RNA, but also to other RNA molecules, including non-coding RNAs, microRNAs, or ribosomal RNAs. So transcriptomics deals with all of these different RNAs. But for bulk RNA-seq, and I guess the classical idea of transcriptomics, what we're looking at is the expression of a gene. So that process of converting, of taking DNA as genetic information and translating it into a functional protein. Transcription lies at that intermediate step. So we tend to use it as information both about what's happening in the DNA, like DNA to RNA process, and also as a possible indicator for what may be downstream at the protein level. So transcriptomics are very widely used in many biological experiments, and it's in pretty much any kind of cellular or molecular characterization experiment will tend to see RNA-seq. So in recent years, we've gotten to a point with the technology now that we can get a higher resolution with transcriptomics. So the transcriptomics, as I discussed prior, as like bulk RNA-seq is just measuring the transcripts from kind of a heterogeneous mix of cells. So a tissue sample where we don't exactly know the specific composition of it, but we're just measuring the counts of transcripts that we see in that entire sample. But as I mentioned, we know that from cell to cell, gene expression activity is going to differ, especially depending on the cell type or other environmental factors around the cell. So that's why single cell has become so valuable now. So with single cell transcriptomics, we can characterize the gene expression from just one cell at a time. And then that gives us insights on differences between cell types, how different cell types might interact with each other and interact with their environment. And then kind of a related idea is this idea of spatial omics. So we're measuring gene expression instead of just agnostic to the localization and tissue architecture that's in the sample. We're now mapping those transcript counts to different regions of a tissue. So it's basically spatial information that we're encoding along with our transcriptomics data. So I guess moving towards the future, this combination of single cell and spatial transcriptomics, which we can also do for some epigenomic measures. So like there is single cell ATAC-seq as well. And then single cell proteomics is another developing area. So the combination of all these multi-omics observed at single cell or in a spatially resolved setting will be very powerful in the future for getting kind of that full comprehensive picture of what is happening inside some biological system. Okay, so that's transcriptomics, and then so after transcription, the RNAs are translated into proteins, and then proteomics is really where we're now studying kind of the functional activity of these proteins that are I guess driving cellular processes. So proteomics is going to characterize not just the levels of proteins, but also their state in that system. And so this is something that is highly dynamic. And I guess just as a rule of thumb, as we go from bottom up genomics to transcriptomics, proteomics, and then the later omics will discuss, things start to change more quickly and they become more variable and are maybe closer to the phenotype, the ultimate phenotype that we might be observing about an organism. So proteomics profiles, these also differ greatly between tissue cells and even within subcellular compartments. So the proteomics profile of the cell membrane is going to look very different from what we see in the nucleus or in the Golgi body or other compartments of the cell. So that's one area actually of proteomics that's emerging, is this sequencing-based proteomics that the idea is you can sequence peptides and then localize them to specific areas within the cell. So it's even more highly resolved than like a spatial approach. And then so the coverage and resolution of proteomics has been improving consistently over the years. So we're much better at it now than we were, I don't know, 5 to 10 years ago. But it is still, I guess, lagging behind in terms of how many proteins we can quantify and to what sensitivity we can measure them compared to transcriptomics and genomics. And then currently, the most widely used technology for quantifying proteins is mass spec. So how this works is we take where we denature the proteins, take proteases, and slice them up into smaller peptide sequences, and then we run those peptide sequences through mass spectrometer and get spectra for these individual peptide fragments. And then we try and map those peptide fragments to the proteins that they originated from. And that's how we get these spectral counts that tell us how much of the protein was present in the sample. So there's a lot more challenges with this because we're taking spectra and from peptides, not from the full proteins themselves. And then we also have to map those peptides to the proteins. So there's a lot of I guess steps involved in this quantification where that introduce some noise in this process. And then mass spectrometry also tends to be biased towards those highly abundant proteins. So these are still some of the challenges that we're working through when it comes to protein quantification. But an alternative to mass spec that is maybe more precise with which proteins we're identifying are these antibody-based techniques. So we have a specific antibody that we know binds to a certain protein, and then we can use different assays or staining techniques to then measure how much of that protein is in the sample. But some limitations with those approaches as opposed to mass spec is that, first we require these high quality antibodies to already exist. So for new proteins or maybe proteins that just antibodies we don't have them yet, those we would not be able to measure. And then often these antibody-based techniques often lead to some qualitative measure of protein expression. So an example is the human proteomic atlas. They use immunohistochemistry staining for protein quantification, and it requires some manual annotation of how much of the stain is observed in an image. So it's not as quantitative as mass spec. But in general, all of these proteomics measurements are important, and we've kind of seen that over and over again now, because they provide a lot of value in addition to that transcriptomic information. So we know that in the process of translating a messenger RNA into protein, there's a lot of modifications that are happening, a lot of I guess regulatory influences from other systems in the cell. So there's most definitely not a direct correlation between the transcriptomic abundance and the protein abundance. So that's why looking at both proteomics and transcriptomics together has been really helpful in, I guess, elucidating some of those the relationships between RNA and protein, and giving us more information about these signaling pathways, and how like messenger RNA and protein, this process of translation is regulated. Okay, so we're related. I guess this kind of falls under proteomics, but it is a post-translational modification. This is phosphoproteomics. So phosphorylation is a very important regulatory and signaling mechanism that proteins use. So a kinase carries out this process, and it takes a phosphate group from ATP and attaches it to some substrate protein. And it's a reversible reaction, so you can phosphorylate and then dephosphorylate a protein. So phosphatases are responsible for the hydrolysis and dephosphorylation of a protein. And it may be the case that the same kinase carries out both those reactions. So a kinase and a phosphatase are not necessarily two different enzymes. But phosphorylation is super important for a lot of cell signaling mechanisms. So a very prominent example is in cancer. This dysregulated autophosphorylation of EGFR is, across many cancers, is a driver for those downstream pathways that lead to uncontrolled tumor growth and survival. So yeah, phosphorylation as a signaling mechanism tells us a lot about how these proteins are communicating and interacting with each other. And how we measure phosphoproteomics is also a mass spec based approach. So the same way we do proteomics, we'll quantify or take the peptides and then quantify them through mass spec. And then to specifically get just the phosphorylated proteins, there's an enrichment step through chromatography that's also added in. And so basically we use like these metal ions that are strongly attracted to those negatively charged phosphate groups to fix them, and that kind of helps filter out the rest of the non-phosphorylated proteins that we might not be as interested in. So there's a lot of challenges with trying to study phosphoproteomics. The first is that phosphorylation, as I mentioned, is this reversible modification, and it's highly dynamic. So studies have shown that within seconds to minutes, which is maybe as long as it will take you, or shorter than it will take you to collect the sample, and then run the mass spec and all of that, it changes at a very short time scale. So it's very possible that what was in fact happening in the cell at the time of sample collection is maybe not this identical to what you've observed from mass spec. And then another challenge is that the phosphoproteome makes a very, very small fraction of the proteome. So the technology has to be super sensitive to just this 0.1% of proteins that are phosphorylated. And they also come in a wide range of abundances. So there's a lot of very abundant phosphorylation events, and also a huge portion of phosphorylation events that are meaningful but have very low abundance. So with mass spec being in a abundance biased quantification approach, that's definitely a challenge. And then a third challenge is that a single protein has many phosphorylation, or it can have many phosphorylation sites. So when we say that one protein is phosphorylated, if it has other sites at which it can be phosphorylated, then we need to understand how those different sites interact. And so the full picture of those dynamics can get very complicated. But nonetheless, phosphoproteomics is a super valuable source of information for understanding protein signaling and protein interactions. Okay, so now we have glycoproteomics. This is another, branch of proteomics that focuses instead of on phosphorylation, the glycosylation of proteins. And one thing to note is that although glycoproteomics is, its own omics, glycosylation also can happen to lipids, RNAs. So it's not just proteins that receive this modification. But glycosylation, is covalent attachment of a carbohydrate group to proteins. And this is really important in a lot of cell communication. So how the cell interacts with the surrounding extracellular matrices or surrounding cells and other features of the environment, and then how it interacts with pathogens and how it stabilizes relative to the architecture around it. So most glycosylated proteins are found at the surface of the cell, enabling these communication-related processes. But they're also very important in a lot of disease pathophysiology. So another cancer example is the glycosylation of MUC1, which is this epithelial antigen. In many tumor types, a high glycosylation of this antigen, it enhances immune evasion and then leads to cancer cell proliferation signaling. So it's sort of helps the tumor survive in its environment. So this is one example of why glycosylation as a process is important to study. And then how we measure glycosylation is also using MS-based quantification, at least for the proteins. So similar to phosphoproteomics, glycopeptides are also quantified by mass spec. And then there's a variety of chromatography approaches depending on the size and then the characteristics of the glycoprotein. You can use different extraction methods to sort of separate out the glycopeptides from the other peptides. But it's also subject to many of the same limitations that we mentioned for phosphoproteomics. Okay, so the last set of omics we'll go over are metabolomics and lipidomics, and I kind of group these together because a lot of what lipidomics focuses on is lipid metabolism, which falls under metabolomics. So metabolomics in general, it is this characterization of all of the metabolites within a biological sample. So after discussing proteomics, which focuses on protein state and function, metabolomics is characterizing what happens after that. So after the proteins and enzymes have carried out the chemical reactions that they're responsible for, and so these metabolic processes or other reactions that are essential for cell survival, metabolomics is going to measure what's going into those reactions and what's coming out. So by looking at the metabolites, we can kind of get an idea of those chemical reactions that are happening and the actual function of those proteins. So lipids are especially important because they play such an important role in cell membrane structure and signaling pathways. And a lot of lipid metabolic alterations have been closely linked with different diseases. So an example where metabolics and lipidomics are both really important is type 2 diabetes, which is characterized by elevated levels of these carbohydrate metabolites, so glucose, lactate, and ketone bodies, and then altered levels of these citric acid cycle intermediates, which those altered levels that we observe reflect some kind of mitochondrial dysfunction. So that's one way that metabolomics can be useful for understanding some disease process. And similar, so yeah, these sequencing of or quantification of these larger molecules is primarily done with mass spec again. So metabolites and lipids are also measured in this way, where they're first separated out by some chromatography method and then measured through mass spec. So yeah, metabolomics and lipidomics, these lie kind of most closely to I guess the organism's phenotype in terms of this pyramid of multi-omics. So we started out from genomics, which is originating at the genetic material level, and then we've worked our way up to how that information is carried out into proteins, and then the chemical reactions that they carry out, which then determine kind of the overall phenotype of an organism. So those are kind of all these layers of interacting molecules that we've discussed.